Welcome to another edition of the bioRN Member to Member session. We are streaming live from the bioRN premises today. And I'm Stephanie. I'm very happy to be able to interview an exciting member of the bioRN network today. As biomanufacturing continues to evolve, driven by the rise of complex therapies like cell and gene treatments, there's a growing need for enabling technologies that go beyond traditional solutions. Innovations that enhance process control, ensure gentle handling of sensitive cells, and allow for seamless scalability from lab to GMP production are becoming essential. Whether in stem cell expansion, viral vector production, or other high-value bioprocesses, the ability to maintain cell viability while scaling efficiently can make the difference between promising concept and a viable therapy. This is exactly where BioThrust comes in. As a young and dynamic company within the bioRN network, BioThrust is developing an innovative membrane-based stirring technology designed to meet the specific demands of modern bioprocessing. It all began with the production of detergents based on renewable raw materials that produce less foam. Sometime later, the company won several awards for its innovation, thereby attracting the attention of investors. Well, I don't want to explain more because I have my interview partner here with me today. A warm welcome to Konstantin Kurz. Great to have you here.
Thank you. Thanks for the great introduction and really happy to be here.
Wonderful. So, as a warming up question, Konstantin, please introduce yourself and how you became part of the company.
For sure. So I am CFO and co-founder of BioThrust. I am responsible for all business development activities here at BioThrust. I have a business background, so I studied business administration at WHU, close to Koblenz. Then went into strategy consulting with the company Oliver Wyman. Been there for 1.5 years and realized I want to do more with startups. And they were usually not the typical clients of such a big consulting company. So I started doing a PhD at the Technical University Darmstadt in the field of entrepreneurship, researched about startups, but also had the intention to join such an innovative startup, and that worked out well with my nowadays co-founders Patrick and Moritz. I met them digitally through co-founder matching, and now here I am.
And it's a perfect match between science and, let's say, economy somehow.
For sure. So Patrick and Moritz, they have a background in engineering and biotechnology, and I think this really makes the uniqueness of BioThrust. So it's really important to have these different disciplines coming together to really achieve deep tech innovation.
So now we want to come, of course, to the story of the company. When was it founded? By whom? You already mentioned, but what was the motivation and the vision at that time?
Actually, it all started long before I joined. So back in 2017, my co-founder Patrick was working on his PhD, and he had the objective to develop a process for sustainable manufacturing of biosurfactants. And as you can imagine from your washing machine at home, these detergents, they really love generating foam. And this particularly happens if you put them in contact with oxygen or with bubbles. So the process required oxygen, and this was traditionally implemented through traditional stirred tank bioreactors with so-called sparges. So you would put in the oxygen into the system, and this led to a lot of foam formation. So you must imagine just for a working volume of 250 milliliters, which is close to the working volume of that drinking glass here over here, they needed like 40 liters of collection tank.
Wow, yeah.
So that showed Patrick and the colleagues this will never work industrially, and it's not meaningful to continue that. So he came to the idea, let's do a bubble-free equation of bioreactors. And this sounds really simple, but it's actually a paradigm shift. 3 years later, they developed the first prototype, and they used membranes for doing so, because his PhD supervisor Matthias Wessling from the RWTH Aachen University, also here in Germany, uses membranes to solve everything, and he's one of the worldwide leading membrane researchers. So that's how they solved the issue. They filed for patent, and they got really interesting and strong interest from industry, also from local partners, but also from the bioprocess industry in general here in Europe. So they realized it must be something that can be commercially used, and I think that's also what was their most important vision. So they want to get their work and the prototype somewhere out in use and not dying silently in PhD thesis.
Okay, and you did a product market fit analysis that leads you to, of course, the market you're into right now. So could you tell us a bit more about this development?
Exactly, of course.
Discussions, I suppose.
So people who have taken a look at our website realize that we are not selling biosurfactants or some sustainable detergents anymore, but we are focusing a lot on cell culture, particularly on cell therapy, and we want to enable the cell therapy era of tomorrow. So how did we end up there? That started in 2022. We started to found our company, and we realized we have a really, really great platform technology. So in principle, our bioreactor technology can be used in any bioprocess, and we must find the application where it's best suitable. Mm-hmm. So we did a lot of research, did a lot of product market fit analysis, talked to key opinion leaders, talked to customers, talked to industry, and realized we have a strong value for the cell therapy field, and also cell culture in general, but cell therapy in particular. There's a new era of stem cells, and I think you nicely mentioned it in your introduction. So we must find production technologies to scale stem cell manufacturing. I think that's the best use case we can have, and we have the vision to become the era of the backbone of this new medical era.
Wonderful. And you brought along a great video that briefly and concisely introduced your team and your innovation. So let's start with the video.
We are BioThrust, and we are developing the world's first bionic bioreactor for a new era of cell therapy manufacturing. We are on the brink of a medical revolution, where stem and immune cells are set to treat once untreatable diseases like cancer and autoimmune conditions, impacting millions of patients. In fact, more than 1/3 of pipeline drugs now focus on these cutting-edge cell and gene therapy approaches.
However, there's a huge challenge. Stem and immune cells are very delicate. The bubble aeration used for the oxygen supply in conventional stir-tank bioreactors, which are the backbone of biopharmaceutical production, creates shear forces which damage or destroys the cells. As a result, Big Pharma resorts on stacking petri dishes, an incredibly slow, inefficient and costly method for scaling up stem cell manufacturing.
These outdated manufacturing methods and inadequate bioprocess technologies are the main limiting factors preventing the new cell therapy era. We thus need to rethink bioreactor design to be able to introduce new treatments for millions of patients in need.
That's where ComfiCell comes in. We developed the world's first bioreactor that provides sufficient oxygen supply without generating high shear forces, scalable to several thousand liters. Central to this innovation is our patented membrane stirrer, an advanced artificial lung that enables bubble-free aeration of cell cultivations, thereby significantly reducing shear forces while achieving unsurpassed gas transfer efficiencies, making it the ideal next-generation bioreactor for next-generation therapies.
In collaboration with industry-leading partners, our technology has successfully expanded pluripotent stem cells and achieved the world's first scale-up of complex lung and cardiac organoids to 2 liters. By 2030, we project a need of 11 quadrillion stem and immune cells.
Our diverse team uniquely combines expertise in biotechnology, engineering and business development. With the guidance of renowned advisors from the cell therapy industry, we are strategically positioned to make a significant impact in this field.
We can overcome the barriers to scaling sensitive cell manufacturing and achieve our vision: affordable cell therapies for everyone. Thank you.
Well, thank you for this very convincing and clear summary. And thank you for sharing the story and the journey so far. But before we dive a bit deeper into your innovation, let's talk about the people behind BioThrust. Can you tell us a bit about your team? What expertise and backgrounds do you bring together?
So I think what's unique about our team is that we are like really, really functional diverse. So we are combining expertise in the field of bioprocess engineering, conventional engineering, biotechnology, bioprocesses, and finally also business development. We are, I think, close to 20 people now,
wow.
And roughly of them already full-time, and a lot of students, student employees, and also thesis students. And what's also remarkable with us, I think we have a lot of R&D focus. I think nearly 75% of people at our company are still in the field of R&D.
Okay, so how would you describe your internal culture or approach to decision making so far with, of course, 20 people, from 3 to 20 people in 1 or 2 years?
It's indeed a change, particularly for decision making and for project management. But with all that, we try to keep our spirit of being scientific, fact-based, and always listen to evidence opinions. And with that, I think we can also perfectly grow and encourage employees to raise their voice.
Wonderful. So to all who is listening today, BioThrust could be a potential step in your further career.
Of course.
So now it's time to dive a bit deeper into the product and the key features that set the membrane stirrer apart from traditional stirring or mixing technologies used in bioprocessing. So would you like to give us more insights?
Of course, and I think you might have seen some glimpse on that already in the video, but just to summarize that again. Let's start with the basics of a bioreactor. So usually when you work with bioreactors in cell culture field, you have processes that require oxygen. And to put in the oxygen, you use those so-called spargers. So they have bubbles through which the oxygen is brought into the medium and can be consumed by the cells. It's maybe little similar to the soda stream, if I may use that non-scientific comparison in that regard here. However, these bubbles cause mechanical shear forces. So there are studies showing that every bubble can kill up to 1,000 cells when it ruptures. And at the same time, it also leads to foam formation, as discussed before when talking about the founding story of Patrick. So we need to think of how can we supply the oxygen without having the bubbles. And obviously this is the concept of bubble-free aeration, which is a paradigm shift for the bioprocess industry. It hasn't worked for 3 decades now because no one has achieved sufficient gas transfer. And that's what really is the core of our innovation. We are kind of decoupling the mechanical shear forces from the oxygen transfer. And in that we allow for high oxygen transfer without high shear forces, achieving unsurpassed KLAs compared to our competitors. In the end, that translates to 99% less shear forces and 4 to 10 times more gas transfer compared to conventional bubble aeration.
Okay, so this is also, could you mention something about the principle of geometric similarity at this stage already?
Of course, so I think what's important is we want to have something that's scalable, right? You've seen our vision in the video. We want to be the backbone of the next biomanufacturing era, and we think it must be scaled because otherwise it's too expensive to be used for our patients globally. So we thought of how can we make a system that is scalable up to maybe 2,000 liters, which is the typical production volume currently for conventional biologics. So we tried to have the same, I would say, geometric properties in every scale. And that's why we thought, okay, we need to have this geometric similarity in every scale, achieving homogeneous mixing and sufficient gas transfer across all of these scales. And how do we do that? And probably we can discuss it later, but maybe a first glimpse already is we have the first approach to use a dynamic stirrer for bubble-free aeration with membranes compared to static systems which have been tried before but never achieved sufficient gas transfer.
Okay, thank you very much. Just to let you know, to all the attendees who would like to ask questions, please type them in the chat and I will ask them later in the interview. So now that we've learned a bit more about the membrane stirrer, are there any other insights into the technological approaches, the science in the background of your innovation?
So I think we've touched upon 2 most important things already. The one is we need to achieve sufficient gas transfer, and the second one is we need to have homogeneous mixing, because once you have dead zones, cells will die in these dead zones because the mixing is not sufficient. So these were the biggest challenges we had when developing the system. We used a lot of simulations, a lot of prototyping to get to a system which achieves homogeneous mixing with sufficient gas transfer. But at the same time, you also said we must be scalable. So that's one thing we also had an eye on. And we also had developed a lot of proprietary technology around the aeration system in itself. So we have developed our own single-use bioreactor vessel. We have developed our own drive units with a coupling unit because it's really difficult to get in the gas in a rotating shaft. So that's something we have also patented. And finally, it all contributes to our vision to have the next generation bioreactor platform. So we're not talking about aeration systems only, but we want to have a full bioreactor system single use, which can be used for both R&D applications, but also biopharma manufacturing.
Thank you very much. Sounds really very complicated for me, but still, well. Okay, so how do you validate your systems in preclinical or clinical manufacturing environments? Do you co-develop with partners?
Yeah, excellent question. So I think what's most important for us is to transparently discuss and express with our clients the USP and the value add that our technology can have for them. We see ourselves much as enabler for the therapies that they are working on, for example. So usually I think, as you have seen, we have unsurpassed KLA, so unsurpassed gas transfers, and with that we can achieve two things. One is we can scale up processes that cannot be scaled at the moment because of the sheer forces of bubbles. This is a lot typically the case for innovative cell lines, for example. IPCs are stuck to small bioreactor volumes at the moment because no current bioreactor system works in scaling it up beyond maybe 2 or 10 liters. Mm-hmm. And the second thing is we talk about process intensification, because since we put in more oxygen, we can achieve process densities, so cell densities and process intensities that have not been reached before. So we can make existing processes much more efficient, and we can enable processes that are not yet enabled at all. And I think these two arguments are used when going to customers, and then we offer them low-cost demo ones, so we help them transferring the current process to our system and then scale and grow with the customer.
Okay, so it's a kind of also a consulting process. You go with your
exactly. It's a lot of like bioprocess development, because I mean our system is similar to traditional approaches, but it's still different, which means that we do a lot of like co-process development with our customers, and we have our in-house bioprocess expertise to support, for example, also academians who have not yet been working with a bioreactor to scale it up to bioprocess levels.
Okay, so how does the membrane stir integrate with existing bioprocessing workflows and equipment?
It's our vision to have a ready-to-use platform, ready-to-use system. So we're not only selling the bioreactor aeration system, but we're selling a full bioreactor platform in different scales and different levels, so that our customer can buy the full system and directly start working with it without any further systems needed. Mm-hmm. So we are actually working on our proprietary approach.
And thinking about automation technology, do you already have a software?
I would say a question that is often asked to us, particularly by business guys usually. We are certainly working on that, because software will be an important part of the bioreactor platform once you move into like automation and AI-based features. So that's something we have planned for the next half year. We are certainly having a look on that field.
Interesting. So, and how do you approach sustainability in your technology and operations?
Yeah. So I guess people from the biopharma industry listening to this one here will know that the whole industry is favoring single-use operations at the moment. So there's a lot of plastic use because it's on the one hand it's cheaper, on the other hand it also ensures patient safety. Mm-hmm. So I think what's really important here is that we have a great view on social sustainability, because we are contributing to affordable health, and at the same time also ensure that patients get drugs without any risk of contamination. And that's the value of I would say single-use technologies. Although on first glimpse they might not be ecologically sustainable as you would have with a glass bioreactor, for example. So that's a question that certainly is asked in the industry, and I think there is progress in also working on sustainable plastics. So I think there's something to look at, but that's not something we will touch in short term.
Right now, okay. So talking about the main drivers for the success of your company, of course, we should mention that in January 24 you hit another milestone with the funding of from Freigeist Kapitalfranken. Everybody knows him probably. So how is the collaboration with Freigeist apart from providing financial support? And can you tell us something about how you are being supported so far?
Yeah, so we are really, really thankful for the investment of Freigeist, and I think it's a really, really value add that boosted and accelerated our journey actually a lot. So I think what's unique to Freigeist is the so-called 360-degree support. So they try to not only support you with capital, but also with all non-financial efforts. And that includes support in setting up company structures, sales structures, but also a lot of marketing and HR activities. I think, in fact, last year it boils down to having 1 to 2 additional FTEs effectively, in addition to what they have invested. So I think we are really happy about that support, and also we were really convinced on how quickly they became familiar with our topic, because I mean we're really a deep tech company, so it's not something really basic like in other business models, and I think that's really something that has set them apart when also talking with us in the due diligence.
Right, and you can focus, of course, on the main topic, your innovation, your product.
Exactly. So it helped us a lot to focus on the really, let's say, important business functions and get the supporting functions also supported by FIS.
Great. So today we are also actually here in a bigger context. BioThrust is part of the bioRN ecosystem since a while, and even before you have been cooperating in the region very intensively. Would you like to comment on this? Why do you like, or why do you love Heidelberg and the Heidelberg ecosystem?
I think Heidelberg is really one of the most vibrant startup scenes currently in Germany. I mean, you have been renowned as I think #1 startup city in Germany last year or even this year. And at the same time, it's also one of the most important biotech hubs. And I think that also accounts for your bioRN network. I think it's one of the biggest networks. And we are really excited about the conferences. I think we had the first contact point with the annual conference of bioRN, but also with the Life Science Investors Day. And both of them were excellent conferences, and they kind of like attracted us to the region here. And I think that's why we also support the region. We're working a lot with partners in the region and also try to get to the events in the future.
Bringing me to the point that cooperation is of course essential to innovation. So would you like to share an example of actual cooperation going on in the region?
Of course. So I think we're really happy that during the mentioned annual conference we have also met the DKFZ and Mark Thier. And we are now starting an experiment with that group in the field of IPCs, and they're interested in scaling up their processes. We will provide the bioreactor hardware, but we're also supporting with bioprocess knowledge. And I think that's a great synergy of how bioprocess companies like us and leading academians in Germany and Europe can collaborate. And I think it's a really promising application node.
Wonderful. Thank you very much. So now we are coming to the questions from the attendees. And the first question, Konstantin, is I would be an opinion to also do process development as business model. Is this something you're going for?
Yeah, that's a great question, because effectively, obviously, we are learning a lot about how to optimize the processes with our bioreactor, and we are probably the best knowing our bioreactor and the different processes here. So that's certainly something we are offering to our clients switching to our bioreactor, particularly if these clients are switching to the 3D bioreactor system for the first time. So I would say it's certainly an add-on service that we are offering to our partners.
Thank you. And second question, you mentioned scaling. What's the trajectory there? You are still quite far from commercial volumes.
Depends on how you define commercial volumes, right? So I think in the field of cell therapy, commercial volumes can be in the range of 2 to 10 liters even. That's something we're going to reach by the end of this year with our single-use system. And then next year we're going to scale up to 10 and 50 liters. And I think 50 liters will certainly be enough for most early cell therapy applications. We already have a 200-liter prototype. So I think even in the field of conventional biologics, we are, I would say, getting closer to industrial volumes, although not yet 2,000 liters. But if we talk about process intensification, smaller volumes can also be more attractive, actually.
Okay, thank you very much. So now it's time for a kind of call for action to the network. So where are you looking for cooperation? Which kind of members in which topics specifically?
So I think in general we have two groups that we are kind of looking for. One is biopharma and CDMO companies looking or working in the field of biopharma manufacturing, and those aiming for either scaling up thus far unscalable processes or reaching unsurpassed cell densities. I think that would be an interesting partner for us. And the second field is obviously academians working with innovative cell lines, for example IPCs, MSCs, T cells or maybe NK cells, and interested in moving to clinics, interested in scaling up the processes. I think we could be an ideal partner for you. So really happy to support all these types of groups in process development.
Great, wonderful. Thank you very much. So We're coming to an end, but before closing, would you like to nominate a member for the next upcoming M2M session, and why? This member you might mention right now.
So, I have one name in my mind. This is the company PL Bioscience.
Okay.
They are likewise from Aachen. We
Yeah.
are Aachen based, so we know them from early on, and they are also working in the field of cell culture and also cell therapy a lot. So I think it would be quite fun to have them here.
Wonderful. So, PL Bioscience, you're pretty much very much welcome for the next M2M session. That will be approximately at the 15th of July. So we are going to mention that, of course, on our website. You will also find the video of this interview on our website very soon. And with that, I would say thank you very much, Konstantin, for visiting us today. I know you're heading off to Aachen right now with the train. And I hope to see you soon back in the bioRN Life Science Cluster, maybe for the Smart Lab Summit. We talked about it. Okay, so thank you very much, Konstantin.
Thanks a lot for having me. It was a pleasure to be here, and yeah, looking forward to also hearing from the audience.
Thank you, and bye-bye.